Hello, welcome to Crash Course Biology. This is Professor Ayman, and today we will be talking about the DNA replication process. First, let's start off by learning what the components of a nucleotide are. There's a 5-carbon sugar called deoxyribose because it contains one less oxygen. Along with the 5-carbon sugar, there's a phosphate group and a nitrogenous base. In DNA, there are 4 different types of nitrogenous bases. These include cytosine, thymine, adenine, and guanine. The nitrogenous bases are split into 2 groups, purines and pyrimidines. Purines are nitrogenous bases with 2 rings, while pyrimidines have a single ring. Purines are adenine and guanine, and pyrimidines include thymine and cytosine. There are 2 hydrogen bonds that form between adenine and thymine. There are 3 hydrogen bonds that form between cytosine and guanine. The structure of the DNA is a double helix and runs anti-parallel. Anti-parallel means that one strand runs from the 5' end to the 3' end, and the complementary strand runs from the 3' end to the 5' end. The 5' end is always attached to the phosphate group, while the 3' end is always attached to the hydroxyl group. The backbone of a DNA includes a phosphate and a sugar. The DNA replication occurs at the origin of replication. The origin of replication has a specific sequence of DNA. The replications begin with an enzyme called helicase. The helicase breaks the hydrogen bonds between the 2 parental strands. The helicase forms a replication fork. A replication fork is a Y-shaped region at each end of a replication bubble. As the double helix unwinds, the single-stranded binding proteins stabilize the unwound DNA and keep them from repairing. When the helicase unwinds the double helix, there is a leading strand and a lagging strand. The leading strand goes towards the replication fork, while the lagging strand goes away from the replication fork. In the leading strand, there are many enzymes that are part of the process. The primase is an enzyme that adds an RNA primer to the parental template. The RNA primer functions as the base pairs. For the leading strand, there is only one primer needed. It adds 1 nucleotide at a time to the 3' end with a total of 2 base pairs added each time. Then another enzyme, called DNA polymerase III, finds the primer to add DNA nucleotides 1 at a time to the 3' end, which totals to 3 nucleotides added. This enzyme forms phosphodiester bonds between the 5' phosphate and the 3' carbon, and it is continuously made towards the replication fork. Then, the DNA polymerase I replaces all the RNA nucleotides in the primer with DNA nucleotides. This completes the replication for the leading strand. Now for the lagging strand. The lagging strand is anti-parallel, meaning that it runs from the 3' end to the 5' end. The lagging strand is created in fragments called Okazaki fragments. The primase adds the RNA primer to 1 nucleotide at a time, to the 3' end. This creates a total of 2 bases each time, and 2 Okazaki fragments. Similar to the DNA polymerase III in the leading strand, this enzyme will find the primer and add nucleotides 1 at a time to the 3' end, creating 2 nucleotides per fragment. The DNA polymerase III continuously adds it until it reaches another primer. When it reaches another primer, it detaches, creating a fragment. Many fragments are created in the process. These fragments are called Okazaki fragments. Then, the DNA poly I comes and replaces all the RNA nucleotides with DNA nucleotides. The last step is the DNA ligase, which connects the fragments that were formed from the DNA poly III with a phosphodiester bond. This concludes the replication process for the lagging strand. I hope you enjoyed the DNA replication process and learned a lot from this video. Please subscribe.